master genetic code safely inside the nucleus of this cell. Can I make as many copies as I want? Sure I can. That's why, what do we allow to leave the nucleus is the what? The messenger RNA goes out there. If the messenger RNA gets damaged or gets messed up, who cares? I can always make a what? Another copy. You get it? You see why we don't ever let the DNA go out there? The DNA is the master copy. Here's the analogy that I use that I think might make sense to you. You know, if you, let's say that your family comes into a lot of money and you decide, you know what, we're going to build the best lakeside home. You know, we bought the best lakeside property on Lake Massabesic or Lake Winnipesaukee, and we're going to build this incredible house, right? So the first thing you do is you go to an architect, right? And the architect designs, right, all of the plans for this beautiful home, right? And when the architect is done, the architect gives you the master what? The master blueprints, right? How many copies does the architect give you? One copy, right? Here's your $50,000 bill. You give me $50,000. Here are the master genetic blueprints. Now it's the first day of construction, and the guys that are come to pour the foundation are arriving. How many of you would give them your $50,000 master blueprints? Hey, here they are. Here's the blueprints. Pour the foundation. I don't think so, right? You only got one copy of those master blueprints. What would you do?
Copy.
You'd make a photocopy of the entire blueprints? No, only those pages or the page that tells you how to pour the what?
Copy.
How to pour the foundation, and you would give them that photocopy. Because what's going to happen in the first coffee break of that construction crew, they're going to step on them, there's going to be, you know, concrete, right? So they get damaged. Do we care? No, because you very safely kept your what?
Master.
Your master genetic blueprints are in the safety of your office. And they come to you and say, hey, you know, we really wrecked your blueprints, you know, during our coffee break. We spilled coffee on it, we stepped on it, it's all ripped and torn. No problem, I'll make you another what? Copy. Do you see why that DNA is the master code? And that's why it never leaves that nucleus. That's why we have to convert the DNA to the what? To the RNA, because the RNA is not going to stay in the nucleus, is it? It goes out here to the cytoplasm, where the ribosomes will then start reading it, as we did yesterday, right? What's the first triplet going to be that the ribosome is going to read?
AUG.
Guaranteed, it's going to be AUG. And when that ribosome reads AUG, it says, I'm supposed to what here? I'm supposed to start here, and I put which amino acid first? I put methionine, you get it? Now, is it only one ribosome that could read this? You think we're only going to make one insulin protein, or maybe I'm going to make 10 million of them? Could this copy be read over and over and over again by millions and millions of ribosomes? Right? So, let me ask you this question. Where does transcription take place? Where does transcription occur? In the nucleus. Where does translation occur? Outside, in the cytoplasms. Everybody see? Here inside the nucleus, that's where I transcribe from DNA to what? RNA. Then I translate out here, because the messenger RNA is going to leave the nucleus. There are pores in the nuclear envelope. There are holes in the nuclear envelope, and the messenger RNA is going to go through those pores, and then the ribosome is going to start to what? To translate it into one amino acid after another, after another, after another. Does this make sense? Transcription, translation. Here you saw, I gave you a copy of this 2 or 3 days ago. This is what we did yesterday. Right?
Mm-hmm.
There's the DNA in blue. It's double-stranded. Are we copying both sides of this DNA? No, because the two sides don't code the same information. If I'm trying to make a particular protein, I'm going to copy that side. That one's called the what strand?
Template strand.
The template strand or the coding strand. So that's the one that has the information that I want to make into a protein. You get it? The other side, I don't care about it. It has something different information. You with me here? I'm only copying the master template strand. And so notice that this messenger RNA, it's complementary to this strand, not to what? To this one, you get it? You have to know which one you're copying, yes? So if this is adenine, then this became what? Uracil, cytosine, guanine, and now here's my first coding unit called a what?
Trans.
A codon.
Codon.
And I can look that up in my dictionary. It says UGG, UGG. And I don't know if anybody can read it, but it says
Tryptophan.
TRP. There it is. Tryptophan. Yeah.
What tells the RNA to form the way it does so that it copies that specific portion of
I'm not sure I followed your question.
What tells the RNA to copy that specific portion of the DNA?
There are enzymes that are controlling all of this inside the nucleus. Right? And so, if this is what? Adenine, 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 this will be what? Uracil, 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 and U is phenylalanine. There it is, phenylalanine. So I have made you a copy of this dictionary. This time you can have them. Because this dictionary, I think you would agree, is better than the one that's in your book. We looked at it yesterday. Remember where you have to look up the first letter on the side, the second letter on the top, the third letter on the bottom. Not that it gives you a different answer, but it's easy to make a mistake with that one. Whereas this one, I think it's almost impossible to screw up with this dictionary.
In the cell, when the RNA is going between the nucleus and the cytoplasm, is that because of the transport proteins? Is it
Transport proteins would help that happen, exactly.
I like it.
Proteins do almost what? Almost everything.
One more.
So let's look at the third triplet here, right? We've got CCG, so that should be what? GGC, and on this dictionary, it's almost impossible to see that that would not be what?
Glycine.
GGC, glycine. Yes, everyone?
Mm-hmm.
Are there questions? You understand genetic information because you understand that's all that those chromosomes do. They tell me how to make a protein, and this is how they tell me how to do it. Why can't we just use one nucleotide for one amino acid? One nucleotide, one amino acid. Why does the coding, here's my question, why does the coding unit have to have 3? Why not just one? Wouldn't that be easier? There's only 4 nucleotides, and there are how many amino acids? 20, so that's not going to work, correct? So why couldn't we use a pair? If you've got 4 bases, how many pairs can you make out of that?
Two.
The answer is, let me do the math for you, you could only get 16. 16 is still not what?
Twice.
Still not enough to code for what? 20 amino acids, so evolution had to land on what? On what we call a triplet. You need 3 of them. Now, how many combinations can you have with 4 nucleotides in 3 combinations? What's the answer? There can be what?
20.
64 combinations.
64.
There's 64. More than you need to code for what? 20 amino acids, which explains why there is so much redundancy in the genetic code. What does that mean, redundancy? There are repetitions, right? There are synonyms, I called them yesterday, right? If you look at your handout, your dictionary just gave you, look at how many there are for serine. There's 4 over here for serine, and if you look way over here, there's 2 more. So how many codons code for serine? 6 of them. Do you see what I mean by the redundancy in the genetic code? Because 64 is a hell of a lot more than we need to code for 20 amino acids. Do all 64 code for amino acids? No. Only how many code for amino acids? Only 61 do. Why? Because 3 of them are used to say what? Stops. There's 2 stops there, and there's a 3rd stop there. I did write in start. If you look up the only triplet that I expect you to commit to memory, which is what? AUG. Look it up, and what did I write in for you there? It says start. It says to the ribosome, I'm supposed to start here, and I put which amino acid first? Methionine. Are you all with me so far? Now, we talked about mutations. So I want to talk a little bit more about mutations. This is a pig whose nose and hoofs are glowing bright yellow, aren't they?
That's awesome.
Because we actually took the DNA from jellyfish that glow fluorescent yellow, and we gave it to the pig. We have the do we have the DNA ability? Could we make a human person glow, neon pink if we wanted to? We could. We don't experiment with people, but we experiment with pigs. Doesn't seem to bother. It's got a nice big smile on its face.
Very happy face.
Seems to be happy with that glow-in-the-dark nose. There's the ribosome making the what? The brown thing's the ribosome, the red thing's the messenger RNA.
Making the amino acid.
The thing you see in this teal green, there's another kind of RNA, right? We're only beginning to talk about this stuff, but there's the growing what? What do I know this amino acid has to be right here? That's methionine. Always the what? It's always the first one, because the first triplet, whatever it was back here, said what? Methionine, right? AUG. There we go. I'm giving you a copy of this handout, this transparency. This is all about point mutations. What do we mean by point mutation? It's a mutation at a single what? A single point in the DNA. You get it? A point, 1 point, single mutation. They're called micro mutations. What does micro mean? Small, right? One nucleotide. You get it? One nucleotide. Well, that's a micro mutation. What's a macro mutation? Macro means big, like somebody having an extra chromosome, correct? Would you agree that that's not a micro mutation, that's a macro mutation, right? There's 3 or 4 there. Three. People with Down syndrome, for example, they have an extra copy of what? Chromosome number 21. Well, that's not a micro mutation, that is a huge mutation. You get it having a whole extra chromosome. That's not what we're talking about here. We're talking about micro, tiny little mutations where I have messed up only one what? One nucleotide. Are you with me here, everybody? Micro mutations, right? And there are different kinds, as you can see. Now, what you see at the top in blue, blue is always the DNA, the red is the what? The RNA, and the purple blocks, those are my amino acids. You get your bearings here? Of course, you don't have color on yours, right? Although you could use highlighters to color them. What do we mean by the wild type? That's the normal DNA, no mutation, right? No screw up. That was mean by the wild is the natural, right? It's the way it's supposed to be, right? Now, what I want you to look is over here. There's the mutation in yellow. Again, this is why you don't have the colored version, would be nice. But notice what's supposed to be right here, everybody. This is supposed to be CC what right here?
G.
CCG. Did everybody say it? CCG. Well, we have a mutation that says CC what?
A.
CCA. Now, when we convert that to RNA, this would code for what? If that's CCG, that's going to be GG what? GGC, which codes for what amino acid? Glycine. Look it up. GGC, right? GGC. There it is, glycine. Now, here's the mutation. Instead of having CCG, I got CCA. And so what does the RNA look like? GGT. Well, look up GGT. Right? Well, you have to convert it to RNA. And you're going to get GG what? GGU. And isn't it nice that GGU, just by chance, happens to also code for what? Glycine. So let me ask you this question, critical thinking. Critical thinking, I ask you to do this every single day of this class. Will this mutation have any effect on the protein?
No.
Of course not. It's the same what? Same. Now, is it a mutation? Let me ask you this question. Has there been a mutation? Yes, because have we changed the nucleotide sequence of the DNA? Absolutely. So there is a mutation. But does it affect the protein? No way. Because it just happens to be that that mutation does not change the amino acids. Because there are synonyms, you see, because there are synonyms in the genetic code, do you see that some mutations would have absolutely what effect? None. Do you get it? That kind of mutation, and you see it right here, it's called a what mutation?